Hello and welcome to the Chapter 4 Muddiest Points video. If I had to pick the most important point of Chapter 4, other than like specific fact, other than hey, it's about cells, types of cells, parts of cells, the structures and the functions of cells. If I had to pick one part that was like most universal, it would be this one. That when we say membrane in biology, we mean the fluid mosaic model of a cell membrane. It's a phospholipid bilayer with integral and peripheral proteins that get to do all kinds of stuff. And the difference between different types of membrane are the proteins that they contain. That said, the first muddiest point that was asked, and several people asked about it, was lysosomes and peroxisomes. They're very similar. The difference between a lysosome and a peroxisome is that lysosomes contain enzymes that cut things, because the lys is to cut, and peroxisomes contain hydrogen peroxide. But they both achieve the same function. And the best example of that I found here for a different way to look at this is this YouTube video called Epic Lysosomes. I'm just going to play part of it for you here.
Lysosomes are membrane-bound vesicles.
So you see the lysosomes.
The hydrolytic enzymes degrade
vesicles here.
Nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
I really hope that the audio is working on this YouTube video. If it doesn't, type in Epic Lysosomes in YouTube. You'll find this video. Another part of it, skipping ahead in the video, talks about the use of lysosomes and how you can use them to actually digest your food. Here's an amoeba digesting its food. So here we have a vacuole full of food that's going to get broken apart by vesicles. Okay, hopefully some of that video worked there. Lysosomes and peroxisomes, small vesicles full of enzymes or chemicals that break down larger things. That could be used for defense, or it could be used for digestion. The next muddiest point question had to do with the cytoskeleton and the parts of it. Well, the parts of the cytoskeleton are microtubules, which are small tubes, microfilaments, which are sometimes called actin filaments, and intermediate filaments, which are sometimes called myosin filaments. Actin and myosin are the two main filaments in muscle tissue, by the way. They pull on each other. But the cytoskeleton is made of tubes and filaments. They're all proteins. These are large polymers of amino acids. The difference between the proteins being how strong they are. Microtubules are going to be very strong, rigid structures, and the filaments are going to be more flexible but weaker structures. And here you can see different images with the microtubules stained in green of these different cells and the microfibers stained in red, and how they can help this cell take on some really dynamic shapes. The parts the cell wants to be strong, like this, I guess, stalk on this cell, whatever this is, this is a neuron, are going to be very strong so it stays in place and it doesn't move. But the more delicate interactions on the outside can be shaped by the filaments. Here, this cell wants to be strong on the top of itself. Maybe it's protection or so it doesn't get crushed, but it's softer on the underbelly. Maybe it's interacting with something here. But these microtubules can form the shape of the cell and give it structure. We also saw in the video called The Inner Life of the Cell that microtubules, this case represented by this candy cane looking structure here, the microtubule can also be a transport highway where vesicles, these big balls here, are going to be transported by the kinesin protein with the feet and the hands along the cytoskeleton. So the cytoskeleton becomes sort of a highway that vesicle traffic can be sent along. You could view it as a highway or a railroad or whatever metaphor works for you. But the cytoskeleton, in addition to its structural role, giving the cell its shape and its strength, can also take on this transport role, connecting different parts of the cell. Okay, the last question on the Chapter 4 Muddiest Points had to do with DNA and plastids, such as chloroplasts and other plastids. We don't need to worry about the other plastids. Chloroplasts are the only plastid that's identified that is going to be of interest to us. So just ignore the general plastid part and just focus on chloroplast. There are 3 organelles in a cell that would contain genetic information. The nucleus obviously is one, and the other 2 are mitochondria and chloroplasts. And just to emphasize that mitochondria and chloroplasts are both double membrane organelles. They have an outer membrane and an inner membrane. In both cases, the inner membrane is highly folded. We call the folds of the inner membrane of the mitochondria cristae. We call that folded membrane inside of a chloroplast the thylakoid. So just describing similar structures using different words. That way we know if we're talking about cristae, we're not talking about a chloroplast, we're talking about a mitochondria. Both of these organelles, the mitochondria and the chloroplast, contain DNA. They contain genetic instructions to make proteins because they have unique proteins that are located on these inner membranes that help with their function. In the case of mitochondria, it's proteins to help with hydrolysis reactions to break down organic molecules and make ATP. In the case of chloroplast, it's proteins, genetic instructions for proteins to undertake photosynthesis and capture light energy and make sugars. Now, it's a reminder of where this DNA would come from. We talked about endosymbiosis in a previous chapter, where you had a large cell, we called it Big Red, that took in a little cell, call it little blue or little green. And this endosymbiotic theory says that there was a independently living organism, little green, that came into a larger cell and entered into a mutually beneficial relationship. Because this little green cell was self-sufficient in its own life form, it had its own hereditary material, its own DNA. That little green cell that came in became a chloroplast. That's where the DNA came from. So they do have some of their own DNA inside of them still, even though that was hundreds of millions or a billion years ago. There's still some DNA inside the chloroplast. Okay, in quick summary of this Chapter 4 Muddiest Points, vesicles are small membrane packets that are defined by what's inside them. It could be enzymes that cut things, therefore a lysosome, or it could be hydrogen peroxide, and therefore a peroxisome. Or it could be like we saw in the potato cells in the cell imaging lab, it could be full of starch, in which case it's called a starch vesicle. And then the other major point that people seem to have questions about is the cytoskeleton, where it's made of microtubules, microfilaments, and intermediate filaments that give shape to the cell and provide a path for molecules to move. And some of the differences between microtubules and microfilaments are also noted. Hope this was useful as you study for your quiz and your test.